Hey friends, welcome back to this channel, and this video is a sequel video from our series Metabolism. So in Metabolism we have covered quite a big part. We have first started with the carbohydrate metabolism, and then we continued with glycolysis. Gluconeogenesis we have covered. Glycogen metabolism is still remaining. It will come in future. So under glycolysis we have covered anaerobic respiration, and under anaerobic respiration there are fermentation, lactic fermentation and ethanol fermentation. Video link of this particular video focusing the lactic fermentation and ethanol fermentation will be given something around here in the right upper side corner, also in the description box. So the aerobic respiration, we have covered pyruvate oxidation TCA cycle, and today's video is about oxidative phosphorylation. So oxidative phosphorylation, this is basically the last part of this aerobic respiration pathway. Okay, so here we'll discuss about, so these are our main agendas. So electron transport chain, ATP synthesis, how ATP synthesis works, ATP synthesis, chemiosmotic theory, uncoupling agents, how these uncoupling agents are sometimes useful for our physiology, what are the inhibitors of oxidative phosphorylation, and some graphical representation like how the oxidative phosphorylation rate and the ATP synthesis rate will alter or modify depending upon the availability of inhibitors or activators. So let's first start with this electron transport chain. So now the electron transport chain, as the name suggests, that this is not a single component, this is a chain of components are there. Okay, so and since this is electron transport chain, that means this is a chain through which the electrons are transported. Now remember, this electron transport chain, we are talking about aerobic respiration, and until today's video, like we have covered glycolysis, pyruvate oxidation, TCA cycle, oxygen is not required in either glycolysis or pyruvate oxidation or TCA cycle. Then why it is aerobic respiration? Because oxygen is required in this last phase of this total metabolic pathway. I mean this total aerobic respiration pathway. Oxygen is required over here. You can see over here oxygen is required to this part. So that's why this is aerobic respiration. And electron transport chain, that means electron will be transferred or transported to oxygen, so that oxygen can be reduced to H2 or water molecule. Okay, so the first point I have written here is moving electrons through a series of electron transporters that undergo redox reaction. That is the electron transport chain. So redox reaction means oxidation and reduction reactions. Okay, these cause hydrogen ions to accumulate within the intermembrane space. Okay, before that, we should remember that this electron transport chain consists of four major complexes, which are very core to this oxidative phosphorylation or electron transport chain. That are complex 1, 2, 3 and 4. There is another complex, that is complex 5, which is known as ATP synthase, is also required for ATP synthesis. We'll discuss about this ATP synthesis in later video. So today our main focus will be complex 1, 2, 3 and 4. And where these complexes are located? These complexes are located in the inner mitochondrial membrane. Okay, so this is mitochondria. This part is the inner mitochondrial membrane. This is the outer mitochondrial membrane, and in between these two there is a space that is called intermembrane space. You can see over here, right? You can see over here, this is the outer mitochondrial membrane, this is the inner mitochondrial membrane, and in between them there is a space that is the intermembrane space. And in this inner mitochondrial membrane, these complexes are located. Complex 1, 2, 3, 4, also complex 5, that is located in the inner mitochondrial membrane. So this is the overall view. So you can see over here that this is complex 1. This complex 1, complex 3 and 4, they are like integral membrane proteins. Complex 5, this ATP synthase also is one of the integral membrane protein, but not this complex 2. Complex 2 is rather a peripheral membrane protein, which is associated within the inner side of the inner mitochondrial membrane. That means we can say that this is located in the matrix side, mitochondrial matrix side of the inner mitochondrial membrane. So there is a difference between complex 1 and 2. The difference is complex 1 can uptake 2 electrons from NADH, and complex 2 can uptake 2 electrons from FADH2. This is one major difference between this complex 1 and 2. Another difference is, whenever complex 1 uptake 2 electrons from NADH, 4 protons are transported from mitochondrial matrix to intermembrane space. Okay, but in case of complex 2, which can uptake 2 electrons from FADH2, there is no release of proton in the intermembrane space. So between complex 1 and 2, we can say that there are 3 major differences. Complex 1 is integral membrane protein, whereas complex 2 is peripheral membrane protein, and which is present at the mitochondrial matrix part of the matrix part, or mitochondrial the face of the inner mitochondrial membrane, which is towards mitochondrial matrix. Hope you got that. And another important difference is, complex 1 can take up 2 electrons from NADH, whereas complex 2 can take up 2 electrons from FADH2. The third difference is, whenever complex 1 is acting, 4 protons will be transported from mitochondrial matrix to intermembrane space. But whenever complex 2 is acting, there will be no proton transport. Okay. So what happens after complex 1 or 2 has uptaken 2 electrons from NADH or FADH2 respectively? They transfer their electrons to coenzyme Q or CoQ. This is a transporter. Okay, we'll discuss about this CoQ later. And from this coenzyme Q, these 2 electrons are transported to this complex 3, which transfer these 2 electrons to another component that is called cytochrome C. Okay, and remember, whenever these 2 electrons are transporting through complex 3, that time 4 protons are also transporting from mitochondrial matrix to intermembrane space. Okay, and this complex 3, these 4 protons from complex 3 is transferred to intermembrane space, no matter the 2 electrons are coming from complex 1 or complex 2. Okay, see, this complex 1 is transferring 4 protons to intermembrane space. Complex 2 is not transferring any protons to intermembrane space. But both of them, either way, they are transferring 2 electrons to complex 3. This is part of the electron transport chain. So 2 electrons are being transferred to complex 3. No matter these 2 electrons are coming from complex 1 or complex 2, whenever they are passing through complex 3, there will be four protons that are being transferred to intermembrane space. Okay, and then from cytochrome C, that those 2 electrons will be transferred to complex 4, which ultimately transfer this 2 electron to half molecule of oxygen, so that the oxygen can be reduced to H2O or water molecule. And whenever these electrons are passing or transporting through complex 4, there will be another 2 protons that are being transferred from mitochondrial matrix to intermembrane space. So we can see from here that if complex 1 is acting, in that case, in the intermembrane space, 4 plus 4 plus 2, that means 10 protons are being accumulated. But when complex 2 is acting and complex 1 is not acting, that times only 4 plus 2, that means 6 protons are being accumulated in the intermembrane space. Now, no matter what, either complex 1 is working or complex 2 is working, one thing is common, that is the proton gets accumulated in the intermembrane space. And this proton accumulation in the intermembrane space produce a gradient in the charge, a gradient in the pH, and that is also known as proton motive force. Okay, and then what happens is, now there is a gradient of H plus ion, and the concentration of H plus ion or proton is more in the intermembrane space rather than the mitochondrial matrix. And now these protons, these accumulated protons, they will try to move back to mitochondrial matrix, and they will move back through ATP synthase, this complex 5 or ATP synthase. And at that time, ADP and PI, that is adenosine diphosphate, and one molecule of organic PI or organic phosphate will join together and form ATP, which is the powerhouse or the source of all the cellular and metabolic energy of our body, of our cell, rather to say. Okay, so let's just see what are the names of this complex 1, 2, 3, and 4. Complex 5, we know the name of the complex 5 is ATP synthase. The name of complex 1 is, as you can see, this is NADH CoQ oxidative reductase. Now, why NADH CoQ? Because it can transfer 2 electrons from NADH, and it will uptake 2 electrons from NADH, and it will transfer these 2 electrons to CoQ. And why oxidative reductase? Because this is an oxidation reduction reaction. Next, we have complex 2, and that is succinate dehydrogenase. This is succinate dehydrogenase. Why? If you can remember the TCA cycle, we'll see that while succinyl-CoA was converting into succinate, FADH2 was produced. Okay, and complex 2 can uptake 2 electrons from FADH2, and it can transfer these 2 electrons to CoQ. So this is known as succinate dehydrogenase, because while production of or biosynthesis of succinate from succinyl-CoA, this FADH2 are being produced. So succinate dehydrogenase, and dehydrogenase is again associated with the oxidation reduction reaction. So dehydrogenase is also one type of oxidative reductase. We'll deep dive into this matter when we'll discuss the nomenclature of enzymes in future. Now the third complex is cytochrome BC1 complex. Why cytochrome BC1? Because this particular complex is consisted of cytochrome B and cytochrome C1 complex. You can also see there is a cytochrome C complex very in close proximity to complex 3. And this is complex 4, that is known as cytochrome C oxidase. In this case, cytochrome C is present, and also cytochrome A and A3 are present in this complex 4. Okay, so this is known as cytochrome C oxidase. Okay, why oxidase? Because oxygen, they can transfer these electrons to ultimately to oxygen, and oxygen is reduced to H2O or water. So this is in total, this is the major parts or major points of the electron transport chain. Now let's see what are the other points. It says the electron transport chain is the last component of aerobic respiration, and is the only part of glucose metabolism that uses atmospheric oxygen. Okay, this is, I think, now clear to all of you. Electrons produce molecular oxygen producing water. Yes, absolutely true. In this case, multiple copies in the inner mitochondrial membrane of eukaryotes and plasma membrane of prokaryotes. Okay, so this point says that this complex 1, 2, 3, 4, obviously they are not present in single, they are not singular. There are multiple copies of complex 1, complex 2, complex 3, and complex 4, so that huge amount of ATP can be produced inside our body, inside each cell of each organism. Okay, and as I have mentioned that this is present in the inner mitochondrial membrane, but mitochondria is present only in case of eukaryotes. So what about prokaryotes? Because we know prokaryotes also undergo this glycolysis and this oxidative phosphorylation and all these things. So what happens? Where are these complexes located in case of prokaryotes? There is only one membrane, and that is the plasma membrane for prokaryotes, where all these things are embedded. Okay, next thing is, common feature of all electron transport chain is the presence of a proton pump to create a proton gradient across the membrane. See, these electrons are transferring from one complex to another complex, but that doesn't contribute to this ATP synthesis. The only one and only thing that is contributing to ATP synthesis is this proton gradient. So this proton pump is very much needed. Okay, so this is common for all prokaryotes and eukaryotes. Whenever oxidative phosphorylation is happening, that means there are some proton pumps involved, and they are generating the proton motive force or proton gradient. Electron transport chain of prokaryotes may not require oxygen, as some live in anaerobic condition. Obviously, there are some anaerobic bacteria which lives totally in anaerobic condition. Some of them cannot thrive under in presence of oxygen. So in those cases, the oxygen is not a very potent requirement for ETS. The only thing is we require oxygen as the ultimate or final electron acceptor. So in case of anaerobic bacteria, there may be something other, like there may be sulfur, there may be iron, or there may be some other inorganic compound can act as final electron acceptor. Okay, like if this is instead of oxygen, if this is sulfur, then instead of H2O, H2S will be produced. So sulfur producing bacteria, they produce this kind of things. Okay, methane, everything. This all thing can be used as the final electron acceptor if the bacteria or the organism is anaerobic organism. Okay, so this is it for today's video. I think this makes sense, and in next video we'll discuss about ATP synthesis and how ATP synthesis works. So about this complex 5 we'll discuss in the next video. If you find this content helpful, please make sure to like, share, comment and subscribe. Follow for more. See you in the next video. Bye.